in terms of following up from what Robin just said, there's a host, a plethora of mathematics that goes into, mathematics and statistics that goes into taking that sequence data, analyzing it, putting it into a form that we can then reconstruct trees from. And of course, at the very beginning, I have to say we're all lying, right? And we're going to talk about phylogenetic trees, and that's not one, but it's a really pretty tree, isn't it? Yeah. We will be talking about the types of trees that Robin showed us, and that's already a presumption that we can recognize and encode the information in terms of evolutionary history by something that's tree-like. Maybe we have to say a little bit at the end about something that may be more network-like, but it's not a bad lie. It's true in many good cases. We'll also be thinking about species in this context, just mentally to do that, but really what happens a lot more often is you're looking at gene sequences, and so those have to be stitched back together into species trees. But okay, put something at a level where it makes sense to you, and we'll probably use the word species throughout as we're talking about these structures. So we're going to talk about, in that sense, of reconstructing phylogeny through molecular genetics. I'm already claiming it's an interesting and worthwhile endeavor. I'll give you, following up on Robin, a few more examples of that. Lots of different ways that obtaining the phylogenetic tree involves mathematical and statistical models. We were going to choose one path through this very large field. And what's going to be my take on things today and how this sort of fits into an algebraic perspective are the ways in which a kind of algebraic geometric perspective can inform the process and attempts to analyze and understand these evolutionary relationships through trees. So, well, there's a big tree. There's the tree of life, right? Millions of dollars have been devoted in NSF money and other funding to try to help us locate, understand, determine how all species in this particular case are related. Now, the methods we're going to be talking about today are so-called distance-based methods, and again, I'm only going to talk about a teeny subset of those. One thing they're particularly good for, even in the modern era where we have a lot of computational power, is for working on very large trees. So I'll make a few comments more about that as we go along. If you're already familiar with some other types of methods, why the ones we're going to talk about are still good ones to have in your back pocket. That said, I remember when Raina gave her intro talk the other day, you mentioned one of these things that we do is we tell students there are these terrific applications out there, and then what do? We do some toy example or some non-example. Yeah, that's what I'm going to do today. No, I'm not doing that. I'm not going to do insects. That's another. There's all sorts of large trees. So we're going to play with a small tree in order to understand what we're doing. Okay. So we could make it smaller. Just real quick to point out, there are two essential features for these trees. Maybe there's three. Of course, they're encoding the evolutionary relationships at the level where if you have two that have a common parent and they're the closest, we'll use two types of terminology. Sometimes we might say sisters. We're going to also say cherries, because if we think of things on a tree, how do cherries look? I should be using the red marker. Okay, you have two little cherries that come off of a stem like that, right? I'm from Michigan, so we see this all the time. It's a good time of year for cherries. Also good in here. Now, in terms of the topology of the tree, right, those types of relationships are what are being encoded. Branch lengths will also matter. That, when we have that information, is telling us something about the nature of mutations that are representing the sequences that are representing these species. Sometimes we have that data, sometimes we don't. As we go along with our methods, sometimes we'll pull those things apart. Determining of the topology, so what are the, just who's related to whom most closely, that underlying part of the tree, versus how long ago those things happened, what is the evolutionary distance in that sense, okay? Sometimes you can do things together, sometimes you can't. So the input of aligned sequences that Robin mentioned before, for example, a type of question you might be interested in is in this very interesting family of flora. Mm-hmm. Flora. Yeah. Okay. So these are the Rafflesia. These are in Thailand and in Malaysia. I work with a biologist, Todd Barkman, at Western Michigan, and he does research on these plants. They grow to be, if I read something like 10 feet, I forgot if it's 5 feet or 10 feet in diameter. And when they bloom, they smell like rotten meat. That's how they attract flies, which actually pollinate them. It's like, yeah, I'm like, Todd, you travel overseas for that? He's like, yeah. Okay, so one of the things that you might think about, and he's taken a look at as a plant biologist, is what type of evolutionary relationships might you expect when you have something that's a plant like that that's huge, right? So over time, are these sort of plants which have evolved become huge? Are the other elements in the related families, and these are all sequences for related families, are they also large? Is that something that sort of happened over time? And being stinky and attracting bugs, there's more in there that I just don't know. So what we're going to do in this process is I'm going to tell you a little bit about how the whole picture works, and then I'm going to black box the rest of it, because I can only talk about a certain piece of it. In the exercises that you'll get a chance to do, you will have opportunities to come back and say, no, this is the part I found interesting. I want to know how you take that raw sequence data. I want to have some software walk me through how you process that, how I can obtain my own sequences to show with students, and do something that I actually could do as a mathematician, which is somebody's got a box well enough built that the non-knowing people can actually use it. And then the real biologists who are out digging in the desert, you could show us how to do it step by step. I'll come back to desert and soil microbes tomorrow. Okay, so you take this raw DNA sequence data. You have to, quote unquote, align it because you want to talk about parts of it that are homologous, things that are describing similar things. It's no good comparing parts of the DNA sequence for one species with another part over here for totally unrelated functions. Now, of course, part of it is you may not know what the function is in the first place. So guess what? There's mathematics that helps you do that, too. So this is a popular program, should be Costal W. This is an old printout, but it's aligning, so to speak, that Rafflesia sequence data. So again, some of the tutorials and things that you'll run through will take you through something that's as sort of as elementary as BioQuest, which is a nice BioQuest module. You can let your students get their hands on it, okay? And start doing some of this to more sophisticated processing yourself, if that's what you find interesting. Because it is interesting. Okay. Am I talking fast? Yeah.
Can you say something about BioQuest?
Oh.
Rhonda would like to say with that.
Where?
No, Sam. You are there. That's why you're like nodding great things.
Yeah,
I was like, I know we've got somebody who can speak well about that.
To say a few words about BioQuest.
Yes.
Sure. BioQuest is a 27-year-old biology educational forum consortium that is working to empower faculty to use more computational tools and real data to engage students in scientific problem solving. And we got into personal computing pretty early. So we've been through several phases, but some of those have involved a lot of software development, much of which is archival quality. But there are, you mentioned the Esteem collection, which is a collection of spreadsheet models and tools for mathematical analysis of data. And there are some tools in the Esteem collection around multiple sequence alignment and other ways to analyze sequence data.
So there's a nice little piece. And again, this is one of, I'll tell you about 4 different possible activities, and that can be one of them that we do today. I put together a kind of tutorial that wraps together sort of starting with that piece and then going again to some more sophisticated, I should say, software, not meant to just be tutorial, but still accessible to, say, mathematicians. Okay?
How do you sign up for that?
It's all pre-created with create complex.
You create an account and that's that, and you have full access. And if you Google BioQuest, it comes up right away, so you don't have to remember that.
But when you use it, you should maybe make a comment like, and thank you, Sam, and others for devoting your life to this. So, that would be nice. Little attribution there at the bottom of the page. Okay, so one way or another, you're going to have this aligned sequence data, and one way or another, you're going to have some type of method, program, whatever, for processing it and producing a tree. This is the output of one particular such. I'm not going to use that one today. But it produced this phylogenetic tree, and for example, you can see that there are very small, these are representative of the size of flowers. There are very small flowers related to very big flowers. So this is fairly interesting, right? So the query about sort of comparing size and how that might or might not be a function of evolution or evolutionary history in terms of what's happening most recently. You certainly don't see that the larger ones have only developed recently, because some of the large ones are a bit older, and you certainly don't see that the closest sisters that you have similar sizes. Okay, so many interesting things here. the type of studies that you can take a look at. For our purposes, we've just been thinking about trees that were rooted, right? But for mathematical reasons and biological reasons too, we're often going to work with unrooted trees. Several reasons. One is that generally, you don't know what the root is. Anybody know the root of the tree of life? Which thing was it? Eve. Which one is Eve? We'll come back to that. But you can always, in a sense, you can always, if you're not trying to do everything in the world, you can always throw a root in later. So if I have 4 species I'm interested in, and one of them has nothing to do with fish, I can throw in an extra edge and a node that will be fish, and I can turn one of these into one that's rooted. Okay? So you just add it when you want it to be there. The other thing is that a lot of the mathematical theory, at least some that I'm going to talk about, or that's underlying what I'm going to talk about, reduces to knowing what happens for certain small trees, sometimes just what happens to quartets, which is what you see on the right, 4 species unrooted trees. And so you can get pretty far with understanding and examining what happens on small trees. Robert?
Assumes to be going on with the bifurcation points. Is that an original species that keeps going on mutated?
It's speciation in that sense, right? At least at the at the species level. Right.
So does that mean the original species is gone and then 2 different versions on? Or does it matter?
No. It could be that you. Yes, right. It could be somebody continues and something new is formed. In fact, that often happens. Yeah.
The answer to that is it depends.
Yeah, it all depends.
It depends on the tree. It depends on what you're looking at.
Yeah.
So both answers are equally valid. The original may continue, and you see different deviations off, but you may also see 2 different things. Depends on the tree.
I think biologists more often than not begin an answer with it depends.
It depends. And what you see over here is the real it depends, because this tree is unresolved, right? This is where you simply don't know whom is related to whom, the star tree. Once you split off, though, in a case of 4 species, once you split 2 of them off, then the other 2 are forced, of course, in that sense. So what you see here is for this type of tree, for 4 species, there's only essentially 3 binary trees. Again, binary meaning you have 2 children coming out of a node, right? There's really degree 3 in the sense of 3 edges being linked together. And for Robin's sake, that's an edge. That's how we use that word. Let's see. Topologically, are these different?
Depends on the topology.
They're graphs. In terms of describing the hereditary relationships, if I think in that way, are any of these different? No. When you look at the last one, 3 and 4 are still sisters, and then 2 is the next from whence they come. Over here, 3 and 4 are still sisters, 2 is the next from whence they come, or they're the most closely related. Actually, 2 isn't from whence they come. Same thing here, right? So part of the things, one of the things that you can do as another set of exercises that will be from our book. I have my copy downstairs. Do you have a copy with you? Okay. We'll get to walk through some aspects of, from this one, chapter 10. You'll get to walk through some exercises that examine aspects of the basic combinatorics of these trees, a little more detail about what we mean mathematically when we say something like these graphs are quote-unquote isomorphic. But for our purposes, they're representing the same evolutionary relationships. Okay? And again, at this level, I don't have branch lengths mentioned. Right? Branch lengths aren't a factor here. So it's only, again, who's related to whom, not anything about the evolution over time or the amount of mutation between for those sequences. So at that level, there are ways we describe these, quote unquote, being the same. Okay? And we can formalize that. And so again, you can have a chance. One of the things you could do is work through some exercises on that. In particular, you could examine why and show why there are 2n minus 3 double factorial rooted binary trees, 2n minus 5 double factorial unrooted binary trees. Everybody know what a factorial is? So I'm like, you know. So this piece, And then, okay, so 8 factorial is that way. 8 double factorial, you skip. Okay? So if factorial is like skipping 1 down and continuing to write that product, double factorial is skipping 2 down. So that means if we want to double check that this number is okay, if I've got 1, 2, 3, 4, 4 is the number of leaves / taxa that I'm thinking of here, right? Then 2 times 4 is 8. 8 minus 5 is 3. And 3 double factorial is 3 times 1. And I have 3 different topologies here. Good. Okay. So at least that counting works. So it's a very convenient number to use as you're arriving at this count. Okay? But again, I'm leaving it as an exercise, so I'm not going to prove it. So again, some uses of phylogenetic trees. We already mentioned the idea of classification, ancestral state evolution for a case where we no longer have the actual gene data or protein data from ancestors. Can you infer it? Can you actually even recreate it, which is something that Todd does, is recreated some ancestral genes. For the Solanaceae family, I'll mention that in a minute. Comparative method, like we were just talking about in a way, what do you do when you want, or not what do you do, how do you use phylogenetic information, hereditary information, to inform your investigations at the level of ecology, at the level of comparing species, not assuming that things have arrived on the planet independently, but that there are these hereditary relationships involved. Okay? Protein functional evolution, we use protein and DNA sequences in order to try to infer what the evolutionary relationships are among organisms or among species or among genes. But one of the things you can do is sort of turn it around and say, well, what does the evolutionary history tell us about the potential function of gene sequences that we identify? Right? Yep. Yes? Sorry, I said it's the number, I did say topologies, and that's not correct in the sense of it's the number of distinct labeled trees. So I'm using that term in an incorrect fashion for mathematicians. Yes, that's right. Thank you for mentioning that. No, it's the number of labeled trees, because of course there's really only one total underlying topology. But if I think about the possible numbers of hereditary relationships that could be described by it, of course I need all possible pairs of those 4 elements, and once I choose one pair, it determines the rest. Yeah, thank you. Thank you. Any time on abusing language, please let me know. Not language, please let me know. Start speaking Japanese, do let me know. I was able to do that, but I can't do it anymore. So I mentioned that we're going to be using a very small tree as we think about things. One type of small tree, for example, it comes in as you think about what's the evolution of humans versus apes versus chimpanzees, right? And this is still very hotly debated until fairly recently, and I don't know what's happened with Neanderthals opening that whole can of worms back up. But if you were to take just this part of the tree, 1, 2, 3, 4, 5, so a clade that would start here, that would just be the definition of a clade, is a subtree that starts with one particular spot and continues for all of the nodes, the taxa after that. You would be looking at comparing sequences for 5 things. Okay. Last quick comment again, Solanaceae is a family that includes tomatoes, potatoes, Nicotania, deadly nightshade. If you're feeling threatened, let's maybe go back to petunias. So this is a very large family of great economic importance. And you can look at what's the sort of the function of certain gene families in there, like SAMT, salicylic acid methyltransferase. And that is something that induces both protection mechanisms for plants as well as contributes to their scent. So how does that work? How does that mechanism happen? Okay? There's a lot that's not known about that, and that's one of the things that Todd also studies. Okay? All right, and evolutionary history can help us with that, but it can't help us with things like in Michigan, the tomato potato, which apparently we grow there. Not on Todd's homepage, that's an unfortunate combination there, but come visit us if you haven't been to Michigan lately. Okay, so spaces of phylogenetic trees. Now I'm going to take you into the geometry. All right? What I hope to convince you of, without doing the details on all of these, is that a good way to think about trees is as points. Okay? They specify vectors. They specify n tuples, where the n is going to vary depending on how we encode this data. Right? This is one picture. It's from a very seminal paper by Valera, Holmes, and Boatman in 2001 of what a type of tree space would look like for rooted trees. What you see here is that there's a plane. It's not really a plane, but for this picture, it's a plane. Okay. Maybe actually this one is a plane. Determined by each of these fully resolved trees that you see. Where they intersect, you have a tree that's partially a star tree. It's not fully resolved.
And by fully resolved, you mean that over there?
It's binary, I should say. Yeah, it's completely binary in this sense of we described it before. Yeah, thanks. For this particular type of tree, you can describe them, but what is happening with branch lengths, so all the different branch lengths that could occur and be laid on top of this labeled tree is what would define all the points in each of these areas. Yeah, then that's right. So for a rooted tree, this is sort of the equivalent of a star tree, right? And so that is exactly the origin, where they all come together. When you tack together that space, when you identify things along the trees where they share common lines, planes, whatever, intersections, you get a space that just representatively looks like this, kind of moduli space. My point about this piece is, as we begin to think about trees, and again, I'm going to give you a couple other ways, more concrete, to think about trees. We'll write out what those trees look like as points. As we go to do that, this type of observation is still true. And that observation I mean is this. When you think of trees and you identify them into a kind of space, it's not Euclidean. It's not a 3 space, a 4 space, a regular 5 space. In particular, it's not convex. I can join, if I have a point over here and a point over there, if I would connect them by a line, the things that lie between may not be in tree space. They may not define trees, because the trees are what the white stuff is. What that means, go ahead, Mike. Yep. Yeah, that's one way in this particular format to identify your trees as points.
Every interior edge looking around,
Yes.
that corresponds to a point.
That's right. That's right. So this is sort of...
Every tree you get by shrinking them.
That's correct. That's correct. Yeah, this, unfortunately, this is an unfortunate one in that I don't want to tell you everything about this picture. It's just really good for the idea of tree space, but I'll tell you much more complete picture about a couple in just a minute, doing that translation of trees to points. Okay, this, right? Because I'm not going to use rooted trees in general. Okay. But this factor is one of the things that if you think geometrically, it's one reason why working with trees and trying to figure out what tree matches your data is difficult in a geometric perspective, okay? Because this space is not an easy space to work with. So as we move around this space, and we'll talk about some ways you can do that, you end up having to be very careful. Presumptions that you would make if you were in a regular old Euclidean space, Rn, something like you're used to drawing with Cartesian coordinates, right? Those presumptions won't hold. Okay. So there's a lot of research done in this area I'm not going to talk about today, but those are things I could tip you to some resources for. Okay. Oh, where's my picture? Oh, okay. Well, this is a picture I really wanted to share with you. Okay, so this would be a picture of unrooted tree space. That's never happened before, and it was there before I put it on here. So I'll come back in a second to talk a little bit more about this picture and see if the picture doesn't come back. There's a way to identify the unrooted trees through something that's called the splits perspective, but I think I can live without that for the moment. So you're going to have to keep in mind the kind of old picture here and the idea of moving around trees. Okay. Sorry, that's new. I mentioned when you want to move around in tree space, again, if it were Euclidean, and in some places it is, if you want to think about are two trees close or not, then you might want to use a type of least squares, Pythagorean theorem type of measure, right? Again, that can be problematic because this space doesn't look like a nice space. What you can and often will do, though, is move around the space in ways that correspond to tree operations, so this respects more of the information about the points that the space is made of. For example, there's something called an NNI move. If you can see up here, what you're representing is the idea that it looks like a quartet, but you might have a whole tree at A, you might have a whole other subtree at C, and so on. And an NNI move switches basically. Does this thing have a, this has a little, oh, look, I don't just have to point, I can point with green stuff. So that's C and that B. That's C and that B. What happened here, we switched B and C out from this tree to this tree. Okay, sort of a two swap. That's a nearest neighbor interchange for N and I. SPR move, subtree pruning and regrafting, takes a subtree, takes it off of where it is, and sticks it back on in a new spot. That's what that's representing. B got pulled off and stuck back on there. And there's something else called a TBR move, which also is a type of generalization, higher up level of what you can cut off and the way in which you can reinsert it. So tree bisection and reconnection move. Okay. With these types of moves, you do have a way to be able to treat tree space as something that you can navigate in terms of having a so-called metric. That's like being able to move around in Euclidean space, so relative to trees. So good ways to tell when trees are close and when they're far apart. Any questions so far? Okay. So now I want to talk about some of the common methods, and again, we're only going to focus on a few. There are certainly a lot of methods that are missing from this picture. I've just done some that are representative. You can look at the type of data that you've got and think about how the species or the genes you're examining differ in terms of nucleotide sites. When you list those sequences, how do they differ at site-per-site basis? Okay. Methods called maximum parsimony, maximum likelihood, take a look at that. We're going to focus on a different type. We're going to be looking at ones that you make use of distances. So that's a different way of encoding how the sequences are different. It's a little less direct. There are ones that are clustering algorithms. We're going to have these trees. We have a whole bunch of taxa. How do they come together? How do you know who's related to whom? Okay. You can do it by a type of clustering. We'll talk about neighbor joining a little bit. UPGMA is a less sophisticated version. Those sort of your most elementary thing to do. If you want to play with some examples of that, and a bit of some more I'm going to do. Raina had mentioned this book before, Mathematical Models in Biology by Allman and Rhodes. Has a very nice section that sort of leads you into some of those pieces as well. Okay. So I'll bring that back up. Another way to think about how you're going to take your data and try to determine a tree is through some criterion that you're going to optimize. So even though we said a lot of what we're talking about you don't have to know calculus for, if I use the calculus paradigm, right? You've got some subset. You're looking at a condition for which maybe you can determine if your points or your elements of interest satisfy an optimality criterion. Or can you find a minimum? Is there a condition for which you're looking for a maximum of something? That's kind of what we mean, and we'll go into more detail on one of those. So we'll be talking about minimum evolution in that context. All right.
So more like how close you said some species are?
It's just different ways to figure out what and say how we mean that. I'm going to go into some more detail. Okay. All right, how we doing? You're quiet. That's not good. If you were in my calculus class, you have to get up out of your seats now and walk around. Okay, so this is a long story, right? It's a long story. Before we talk a little bit more about those methods, I'm going to come back to a point that I mentioned already, small trees versus large trees. The distance-based methods we're about to talk about, two different kinds. There's a clustering one, and then there's an optimization, minimization type one. Come out of the neighbor joining method, which is a very old method for coming up with phylogenetic trees. And what this quote addresses is, again, what I've mentioned, a fact that what's more common to do now in this sort of hierarchy is not necessarily to even come up with a single tree, but is to come up with a distribution of possible trees. That's what this Bayesian piece is doing here for. Okay. Maximum likelihood, right? However, again, for very large trees, computational complexity is still a problem. And some of the methods that we'll talk about will let you handle computational complexity because just algorithmically they take a lot less power, a lot less runtime, I should say. Okay. One of the other things that's interesting, there's a recent paper by Stephen Rock. It's recent to me, right? It's 2010. Last time, before I became an associate dean and I was reading all this stuff all the time, that's the last time I remember doing that. He has a very nice paper where he takes a look at sort of distance-based methods. And we're going to see what happens with these methods is we're going to look at distances between sequences in a certain way. What people have been concerned about is that you're losing information when you do that. You're going to wipe out the original sequences. You're just going to sort of see differences between them in a way. So there's been a concern that that's really not going to have the same level of information that something like some of the other methods do, something called maximum likelihood, for example. What his research shows is that's not necessarily true. In fact, we have still more information embedded in some of these methods than we do in the ones that have become more popular now. Okay? So if that becomes something you're interested in, I'll be glad to follow up, and I'll post this paper as well. So here's where we are. Sequence alignment. We have to have some way to turn sequences into data that we can use. That's going to be accomplished by a so-called evolutionary model that I'll briefly describe what that's about. From evolutionary models, we're going to get pairwise distances. That's what we're going to start sticking into our algorithms. That's the way in which we're going to think of species being related or unrelated. Then we're going to have to come up with a way to get the labeled trees and the branch links that go on top of our labeled trees. And there are lots and lots of ways to do all of this. Okay. Okay. So I'm going to flip way back for just a sec. There it is. This is the SAMT gene portion. When you see the sequences up at the top left, CJAT stuff. Right? So again, for each one of the species you're looking at, you've got a sequence snippet. It's been aligned, so it has a nice picture like that. The dashes mean that we've got elements. We did elements we couldn't align. It's all right. Overall, the sequences are aligned. So now I'm going to start talking about how those are different. Now, here's the thing. Just because right now, at this point in time, when you yank a potato out and you pull the DNA sequence from that, right, you see what you see up on there. That doesn't mean, of course, that's the way that it always was. Well, no kidding, because things have evolved. You know that. But what you have to account for is the fact that when you see, say, a T there, it might have been a C a few 100 billion years ago, or maybe only a couple million years ago, right? And then switch back. Okay. So mutations can happen, and then they can come back. There's a sense in which what we have to do is a kind of correction relative to the data that we see right now. Okay, so you don't exactly know what has happened over time. That's part of the point. But to correct for possibilities that, for example, these things, G's might change to C's more frequently than A's would change to T's. There's a number of different possible assumptions that are biologically valid that would cause there to be, cause the idea that if you presume the sequences you see now are what were always there once they speciated, you're being very simplistic. So to control for that, we take a look at a so-called evolutionary model, which what you see here is PAA is the probability that an A mutates to an A, right? Likewise for all of the other terms that you see in here. So the probability that T switched to G. Or it could be the probability that G switched to T. You have to decide to set up the table one way or the other. I'm going to say T to switch to G. Okay. Then, in that sense, you work with a selection of a hierarchy, or from a hierarchy of what are sort of possible parameters that can be involved in the types of mutational switches that you are going to try to, in a sense, control for or be mindful of. You might have a two-parameter model that allows for some variation in sort of two ways. That's a Kimura's 2-parameter model. This one is the Jukes-Cantor 1-parameter model. All of these are representing different assumptions you can make about those probabilities. Okay? And I don't want to go into this in any depth at all, but what is happening is there'll be a Markov process as we think about the mutations occurring, which just means it occurs with some probabilities relative to these, and then it totally forgets where it came from. And if there's a mutation again, it occurs according to these, and then it totally forgets where it came from. And you had a question, yes?
How do you deal with the fact that maybe you can have a mutation, but then you can still get the same protein?
It's in a sense you try to encode that through here. It allows for the fact that that happens. Okay, so what you have to do when you think about how sequences are different is you have to encode, or it's good if you encode, information that respects or supports the idea of whatever you see as differences between your sequences, there's some underlying mechanisms that may have produced those with varying probabilities on the mutations. Okay? So that's what we mean by an evolutionary model. At the end of all of that, we have this sequence data. And if you were to just look at 2 sequences and do the sort of Hamming distance, which just means let's look at where 2 sequences are the same and where they're different. That's a fancy math way of saying that, right? You would tell me these 2 are the same except at one spot. Right? Now, what we do when we build, what we're doing is building the distances between sequences, is we do more than that. Again, we account for possible changes using this so-called evolutionary model. So you're not going to just generally see, oh, my distance between this sequence and that sequence is 1, because they differ by one nucleotide. Right? You're going to see numbers like 3 and 1.8 and 0.6, and these are things that have been generated by those Markov models. Okay? So our goal is going to be this. You've given that sequence data. It's all aligned. You can take all of those sequences and pairwise compare them. Between any 2 sequences, you obtain a distance. Again, it's dependent upon this evolutionary model. Some of the exercises you can do will walk you through what that really means and how you could assess that yourself, come up with what these distances are. Okay? It takes a while. So you're starting off with data from sequences. That data is probably noisy. So even if you believe that there's a tree there, and I already said we know we're lying a little bit, in terms of the noise that comes from your sequences, it probably doesn't correspond, that sequence data itself, to a tree. What would we mean by corresponding to a tree? It would be that there would be a tree that would be labeled by your species, in this case boringly labeled A, B, C, D. And the distances that you get from this process that we've only hinted at between 2 species. What's the distance between species D and species B that comes out of the end, right? Well, what is that distance? It should be, if this tree fits it, 1.5, 3.5, because that's the distance it would take in this particular labeled tree and with those particular branch lengths to get from D to B. Okay? So you will start off with pairwise sequence data and try to recover a tree that fits it in this way. And that's all we mean by tree metric. Okay? The distance data itself, when you associate for these pairs of sequences the distances between them, of course you have something that's symmetric. If you have 2 sequences, it doesn't matter whether you write AB or BA, their distance will be the same, the way these things are generated. Okay. The distance between A and B and C is 0. So in terms of representing the distances, the pairwise distances, all you need is the symmetric matrix with 0 on the diagonal, and all you need is either the upper part or the lower part, because everything else is redundant. You could write it in there, but it's redundant. So that's what this is representing. Everybody okay with that? Okay. So our goal is to come up with, from our noisy data of distances, a tree metric. Okay?
I've just done the measurement
Yes.
of the distance from B to D.
Yes?
Just curious,
Mm-hmm.
yeah,
There.
why is there like a triangle in
There is.
instead of an upper bound?
The-
Some being an upper bound of the distance exactly.
You can define tree metric in terms of basically a triangle inequality holds. That's what would usually define a metric, right? Is that what you're asking? Yeah. There's another condition that will sort of come out of the wash of what we're going to talk about, where a tree, a potential tree, a diagram, will turn out to be a tree when there's a certain so-called 4-point condition satisfied. And that's another piece where you can look at that distance data and ascertain, whoop, that's one of these. It is a tree.
Explicitly that I wouldn't say that the distance between B and D is 3.5.
Why?
I would say that would be an upper bound of the distance.
No, these are meant, yeah, in an actual tree, this is meant to be additive. It is, that's part of our assumption in this case. So when we say tree metric, I'm not thinking of metric on the whole space in that way. It is a symmetric matrix with zeros on the diagonal that satisfies an additivity condition on the branch lengths, on the edge lengths. Yes?
I'm not sure I understand where we're going, but you
Sure.
have that, the distance data, and then you construct a tree, is that?
Yes. And so part of what the difficulty is, a lot of what we will develop when we think about is, well, if you had the tree, what would it look like and what would it mean? Right? That's the real thing. And that's why I covered it up at first. In the beginning, that's all you got. Yep.
And you've got to make a tree that fits that, with maybe many possibilities.
That's right. Hundreds of thousands of possibilities, and the way in which we mean fit will vary. So what we'll talk about in the rest of the time we have is, what do we mean by fit? What do we mean by a good tree in these particular models, and how do we come up with them?
Are there really
Yeah.
going to be lots of possibilities in general, or if there is, will it be unique?
No, in general, and I'll show that picture of tree space again. For these methods, if you start with a tree and you apply the methods, you'll get the tree back. But if you are starting with something that's noisy data, right, you may have a tree that corresponds to multiple different sets of data will generate that same tree. So it's not one-to-one in that way. But there can be more than one tree that will come out of any one of these, well, some of these processes that would still be a valid tree. Yeah.
Will there ever be no tree?
For the methods that we're going to do, no, a tree will be produced. A tree will be produced, which is kind of one of the cool things about.
You can certainly construct matrices like this where there is no tree that realizes that. Are you just ignoring that situation, or?
No, well, so that's the thing, right? And that's sort of the point. In general, when you give a set of these, it doesn't mean that it's tree-like. It's not a tree metric, right? An arbitrary set of, an arbitrary upper triangular matrix like that is not going to be one such that you're guaranteed it would satisfy this additivity property along the edges of some unlabeled graph.
The goal is like to paint a tree metric that represents, but if that's not possible, get something that's as close as you can.
So the reason I said some of the methods we do will produce a tree is it will. Whether what you want to know is that a good tree to represent your data, right?
Yeah, because it's all coming out of the data,
Yeah.
right? So
Something will, yeah. So for some methods, you might not produce something. In this case, we're talking about you will produce something, but is it good? Right. Yeah. Good questions. All right.
Just to throw in a biologist comment.
Please.
A tree that actually lines up and matches the data exactly, I have never seen.
It would be a lie, right? That would worry you. Like when a statistician sees that, I had my base, and then I ran my simulation, and I got exactly the numbers in the baseline. It's like, oh, yeah, didn't, that was bad. Okay. I mentioned earlier, we can think of these pairwise distances because of the nature of the data, if I just wanted to read it off like this, 3, 1.8, 2.5, 2.8, 3.5, 1.3, I could write things in a vector form, in a tuple form, right? So when you have that pairwise distance data from your sequences, you are defining a point, whether you think of it in sort of this block form or you think of it as just a list. So this is a way in which, again, data gives rise to points. Some of the points will be trees, some of them won't. And you're trying to get a good tree at some measure of good. So you're trying to do a projection, you're trying to come up with a tree in this space that is good match for your data. Right? But they're all points. In this case, for these quartets, we're looking at 6 coordinates. Everything's described by 6 coordinates. And when one of my biologist colleagues at Western Michigan said, "You're not going to tell me we're going to play in 6 space, are you?" I'm like, "Yeah, right. This is why this is useful. It's 6 coordinates. I can draw a representative of it, but it's a perfectly good measure. I change around those coordinates. I have different sequence data." Okay. So, yeah, because AA is 0, we'll just do, in this orientation, of course you're going to choose which way you want to write out the matrix elements. But if I start here, I'm just going to work from left to right on the top row. So 3 is there, 1.8 is there. Where did 3 come from? From A to B, 1 plus 2 is 3. So then I'm just going to start listing them like that. And again, you can choose your own order. It doesn't really matter. Just choose something that's consistent and list all your points in that way. Okay. We've got a bit more to go. Are you guys okay? We've been asking questions as we go along, which is part of the point of having a longer time. Is it good to go? Okay, let's get to some more stuff. All right, well, there's neighbor joining. Great. Next. Neighbor joining, which I'll come back and show you. You get a chance to play with that very example that I showed you. Neighbor joining is an agglomerative algorithm. You take those sequences and those species, and you get a way to tell who you should start to stick together. Who are cherries? Okay. How do you choose to do that? If you were to look up one definition, this would be what you see. You use something called the Q criterion. Which essentially takes this type of input data. It causes you to write out some expressions. And you write a new matrix for this type of sort. I'm going to reflect this one. I know this picture is small, but even wherever you are, you can see it's now a lower triangular form instead of upper triangular. The neighbor joining method will tell you how to start with this data, calculate some stuff for every pair, A, B, B, C, D, C, and so on. Get some numbers out. Look at the smallest of those numbers. And whichever is the smallest, if you get an answer of, say, in this case, -13 is a number that's going to occur when you compared A and B for this stuff you calculate, which I'll talk about what we're doing in a second. Whatever you get as the smallest output, that's the pair that you join. And in this particular run of this, and again, it's probably not visible, it'll be when you go to pull things up, it turns out that when you have A and B, you're going to get -13. When you look at the output of D and E, you get -13. So you could join D and E first, or you could join A and B. And when you start off, you just maybe have this star tree, and you don't know who belongs where. There's this idea of an order of so-called cherry picking. The algorithm will let you choose either. You have to choose one in order to proceed. So if you choose A, B, you're going to start to pull those away as a cherry. And then the next iteration, DE, happens to show back up, so you're going to pull those 2 away. It's an iterative process. It each time creates a new matrix of entries for you from which you compute some stuff, and you look for the entries that are the smallest, and you pair together those elements. And at some point, you don't have much left to do, so it's boring. Right. It's really what happens is once we join these two, suppose we join A and B, I'm going to think of a new node, and that's what I'm going to use as an entry in my matrix now, in place of the two things I replaced.
So it doesn't reconsider
Say that again.
the
Sorry.
It doesn't reconsider the use once they're formed.
Oh, it inputs them as elements groups of the new matrix. I'm not used to that. Yes.
As a sharing for A and B.
Then it's
Saves that.
yeah. Then it remains a fixed element that you then play with. Now you might join it to something else. Yes, is that what you were asking? Okay.
When you're faced with a choice,
Yes.
A, B versus D in this case, does the choice in essence matter to the finished product?
The really good part is if I get to the end of this talk, I'll answer you. Okay, it doesn't matter in one way in the sense of you will get a tree out, right? And it will be valid, right? It'll be a valid tree that comes out of this data, and it will still have it will have that distance data as the input. But you could possibly get different things just come out. Yep, that's correct. So I'm going to give you a geometric answer to that question too. Okay. Now, here's the thing. That creation is not unmotivated. Okay? How to come up with the elements that you should join together is itself a geometric, it's a nice little exercise. It's not unmotivated, it's not weird, but when you write it out in a very efficient computational form, it sort of loses its meaning. Okay? Because of the time, what I want to do is just draw a quick picture of one thing, because we'll need that to motivate as far as we can get to the later. I don't think it matters what I call. I'll be sorry I called them these, because I won't think this way. Okay. The criterion that will boil down into this stuff, if I run this on a quartet, which again, of course, the point is to use it for much bigger things, but just to illustrate sort of in a geometric way what's happening, an elementary way what's happening. Okay. What the condition is that you're going to use to minimize is you're going to look at this thing as we go along. I'm going to write out what it is. Pictorially, here's what you're going to do. You're going to, sorry, that's why I said if I label these things the way that I labeled them, I would be sorry that I did. Okay, I might change my labels in a minute. I'm just going to leave it blank for a second, because this is what you're going to do. It doesn't matter what I call them. What matters is it's going to be the effect. So here's what you're going to do. You're going to start here. You're going to run here and go over there. So you're going to take the distance between here and there. Okay. So I've got a cherry over here. I've got a cherry over here. All right. So I'm going to start with this distance. Then I'm going to choose my next one. From this element, I'm going to go to the other element on the opposite cherry. So I'm going to come down here and go like that. So I'm going to add in that distance, which I'm just going to illustrate like that. Okay. And then the next one that I'm going to pull up is, I'm going to do the same thing for the other element in this cherry on the left. So what should that be? I should go here and then there. So I'm going to go like that. Okay. And then I'm going to go here and then there. So I'm going to go up and then down. So those are the distances I'm going to trace out. Okay. Now, if I do that, what has happened here, I've run this middle edge 4 times, right? As far as the other pieces go, I will have run this side and then this side. So I sort of will have run this bit. Then I will have run, same thing over here. I will have run along that edge once. And I will have run along it twice here. This one there, that one there twice as well. Okay. So I'm going to take off, in a sense, those copies that I've run. So what does that leave me with? At the end of the day, what have I traced out? The center edge, how many times? 4, and then if I multiply by a 4th, basically 1. So this is for, if I then am computing the first element of the matrix that I would be looking at, this is, I would start with A, and this would be all of the elements I would add up and create. The lengths that remain, which we just saw what it would be, okay, that's what I'm, and in this version of the method, you're seeing stuff we talked about minimizing. There's a way to change this into something that's a minimization. I'm going to alter it slightly in computation that's more effective. So you can maximize these things or minimize others. Okay? So in that context, what you're going to be looking at the end is something that's trying to control for the fact that just because you might have lengths that are short and some that are long, right, doesn't mean that because that distance is shorter, those two are cherries. Okay. So implicitly, there's an algorithm going on that is replicating at a larger scale this sort of internal representation of these edges. Yes?
Just a second ago, maximizing here versus minimizing there.
Yes.
That sounds to me like a linear program.
There's a whole bunch of linear programming underneath. This is much more direct. It's like this is called, there's one you can call an S or a Z criterion. You change it into, by a very small algebraic transformation, into something that's the Q criterion I showed earlier. That's the thing that's negative. You end up minimizing. For this version, there's something positive that you maximize. It's much more simple than that yet. We come back to that in a minute. More sophisticated way of doing that. Okay. So there are some reasonable ways in which one can join trees. I do this with my eraser in class. I leave it somewhere, which means I left the advancer somewhere. There we go. Okay. Neighbor joining method works very well for big trees, and it's fast. Because these are the types of computations that you're doing. It's very simple and a runtime complexity. Forget if this version's O(n) cubed, O(n) squared, down reduced to that. Okay. Back to the question of what you generate in terms of which trees go to which types of neighbor joining outputs. So there's an algorithm that takes your distance data, tells you how to collect the points, and it spits out a tree. When it does the process I just illustrated, we did this without branch lengths in a sense. So I wasn't focusing on that, but it does produce them, right? And so you will actually get a branch length tree. I just represented some pieces here by this. Okay. The choice of the cherries, when you have a choice, leads to different so-called neighbor joining cones. From spending a little time, which you'll get a chance to do with the exercises if you want, from spending just a little time with writing out this type of condition for what you're calculating to minimize, what you see is you have a system of linear inequalities. That comes back to that piece now. And so when you have a distance matrix, the distance data, you multiply that by a positive scalar, you're going to just multiply, in a sense, the outputs by a positive scalar when you run the neighbor joining algorithm. So you will return, if you have a tree here, you'll get something essentially that's conic. Right? Every time I take a tree and a distance matrix and a multiple of it, I'm going to generate something that's a multiple out from the neighbor joining algorithm. Okay. So neighbor joining cones, you can break your space, your tree space, up into neighbor joining cones. It's a way of thinking about what neighbor joining does. And for a given tree, a real tree, not the distance data we don't know yet, for a real given tree, there are a number of different cones, that neighbor joining cones, that are associated to that same tree. If I think about just your topology, just your labeled tree. And they're not convex. So it's, again, a not nice space decomposition. No, I was just playing around with PowerPoint. Yeah, really just representing that there are these trees. No, it's not a.
Can I ask a summary of the question?
Yes.
So, okay, so if you have a matrix like that,
Yes.
there's going to be multiple trees. So, okay, so every point on that space corresponds to some tree.
Yes.
With the interior edges are some length.
Yes.
Equilibrium points. But it's not a one-to-one map. So there's going to be multiple points.
Possibly. Mm-hmm.
So if you said it's not convex, so if you go from here to here and you work from here to line, what
Yeah.
you're getting, like you're changing the matrix entries in some way.
Mm-hmm.
But in the interior, you're not, you're getting a matrix that's not realizable by a tree.
Right. You
So-
can't just move. That's right, in a straight line.
so
and stay in that tree
so
space.
realistically,
Yes.
you understand that you don't ever get it. So realistically, the data is not going to be tree. So it's going to be somewhere off of this. And what you want to do is you want to like project it onto a different.
Mm-hmm. And so in this certain way, one of the questions that is asked about neighbor joining is, how is it, like, what is it doing in that sort of context? It's a projection of sorts in that you've got a tree, you have this distance matrix in the larger space, and you come up with a correspondence, right? Does it look like, can we describe it in some way that's more familiar to us as mathematicians, in the sense of something from a minimization or an optimization point of view, right?
So what? Okay. So when you do the neighbor joining, you're creating this matrix which doesn't have a realization of an actual tree.
Well, it may or may not itself already be tree metric. You do get a tree out. Neighbor joining produces a tree.
And so the point really sits inside one of these codes, and
Yes.
and you're only going to stay within that code.
There's a union, so,
Because you might have several.
right, because you might have had different ways of picking the cherries, for example. Yep.
The trees sit on the planes, on the surfaces actually, or not in the empty space?
What happens is that, what,
Might be in the,
if we went back to the original picture before we sort of glued things together, right? Points that lay on intersections were trees that weren't binary.
right.
The binary
So
trees,
they were on
yes,
the planes, they were
yes,
on the-
that's right.
And what was in there? So that is the space.
yes.
And so these cones are the, right.
So you might have something in the interior that's not a tree, and the algorithm's going to send it to one of those walls.
Somehow, right. And the somehow is, well, what we saw. That's what it does. Okay. In terms of time, I should be quitting. It's 10:15. I would like to give you an end picture for what we haven't discussed here in terms of wrapping this piece up to something else. So I'm going to do a lot of fast forwarding here, but I'll have these posted. So You sure? Okay. Okay. If I could do that, we'll spend another 10 minutes setting us up for exercises and other things we can do, because I want to tie in this geometric picture with a new way in which we can understand the neighbor joining and others of these methods that has a useful conclusion for biologists. So I'll stop now. We'll take a break.